So today let us discuss about neutrophils, which are the principal cells in acute inflammation. Why neutrophils are the principal cells in acute inflammation? Because they are the most abundant cells in our blood. Suppose we do a differential on our blood, 60 to 70% will be neutrophils. Another reason is they express more adhesion molecules towards endothelial cells. So there are two reasons. One being more abundant in the peripheral blood. Other reason is they express more adhesion molecule. Here is a vessel. These are the vessel lined by flattened endothelial cells. And here we can see some neutrophils. The cells with eosinophilic cytoplasm and multiple lobated nucleus. And this neutrophil reaches the tissue. Here we can see numerous neutrophils with eosinophilic cytoplasm and multiple lobated nucleus. Let us see how these neutrophils are formed. All these cells are formed from the hematopoietic stem cell. They can differentiate into myeloid and lymphoid stem cells. From the myeloid lineage, these RBCs, neutrophils, platelets, etc. arises. Let us see what is the first recognizable precursor of this neutrophil series. These are the myeloblasts. These are the first recognizable precursor of neutrophil, myeloblast. Look at these cells. They have got a large nucleus, high nuclear cytoplasmic ratio, and scant basophilic cytoplasm. Scant basophilic cytoplasm, enlarged nucleus. And here we can see these two white spaces. These are prominent nucleoli. Because these blasts are active cells, their chromatin unwinds and they have got a dispersed chromatin. So high nuclear cytoplasmic ratio or enlarged nucleus. These white areas, prominent nucleoli and scant basophilic cytoplasm. These are the myeloblasts. Then the next stage is promyelocyte stage. This promyelocyte stage is a stage in which the primary granules appear. These abundant eosinophilic primary granules first appears in the promyelocyte stage. The appearance of primary granules in the promyelocyte stage. Then the next stage is myelocyte stage. This moon-shaped nucleus, and then these, in addition to the primary or eosinophilic granules, they have lilac-colored secondary granules. Lilac-colored secondary granules. Okay. Now we have the next stage, metamyelocyte. Here the nucleus is indented, but the indentation is less than half. This is the metamyelocyte stage. In nuclear indentation, less than half. Then if the nuclear indentation is more than half, it is band form, band shaped nucleus. Okay. Then finally we have mature neutrophils with the segmented nucleus, multiple lobes of the nucleus that are connected by these delicate strands of chromatin. These are the mature neutrophils, and they have primary and secondary granule. Let us once again see the different stages. Myeloblast stage with high NC ratio, these prominent nucleoli, scant basophilic cytoplasm. Then the promyelocyte stage in which primary granules appears, large prominent eosinophilic primary granules, and then we have the myelocyte stage with these lilac-colored secondary granules first appear. As we go down, the nucleus becomes smaller and more condensed. Okay, and the secondary granules appears. Then we have the metamyelocyte stage with kidney-shaped nucleus, more condensed chromatin, and the band form with indentation more than 50%. In metamyelocyte, indentation is less than 50%, and the band form indentation is more than 50%. Finally, we have the mature neutrophils with the multiple lobes connected by delicate strands of chromatin, segmental neutrophils, and the primary and secondary granules. So neutrophils migrate to the site of inflammation and they remove the injurious agent. How do they do that? They have got an enzyme called NADPH oxidase. This NADPH oxidase can convert oxygen to superoxide or O2 minus. So NADPH converts oxygen to O2 minus or superoxide. This superoxide can be converted to hydrogen peroxide by superoxide dismutase. O2 minus to hydrogen peroxide by superoxide dismutase. Then this hydrogen peroxide can be converted to hypochlorite by an enzyme in the primary granule. What is that enzyme? Myeloperoxidase. So myeloperoxidase in the primary granule convert this hydrogen peroxide to hypochlorite. Even if the myeloperoxidase is deficient, neutrophils can kill by this O2 minus or superoxide, hydrogen peroxide, etc. But for efficient killing, myeloperoxidase is necessary. So if myeloperoxidase is deficient, what happens? There is accelerated infection because there is inefficient killing. Suppose if NADPH oxidase is deficient, the oxygen is not converted to superoxide and it is not converted to hydrogen peroxide and hypochlorite. So no killing by free radicals. Instead there is granulomatous inflammation, recurrent granulomatous inflammation. So chronic granulomatous disease is a condition where there is NADPH oxidase is deficient. Now let us see the other components in the granules. There are primary granules and secondary granules. Primary granule, in addition to myeloperoxidase, contains other enzymes. For example, defensin, cathepsin, protease, bacterial permeability increasing protein, elastase, non-collagen hydrolase, etc. They all helps in the clearance of these injurious agents. For example, bacterial permeability increasing protein can activate phospholipase, so they can digest the membrane of the bacteria and all. So defensin, cathepsin, protease, bacterial permeability increasing protein, elastase, non-collagen hydrolase, etc. What is the function of this elastase? Elastase can digest this elastic tissue. So this elastase in our body is neutralized by alpha-1 antitrypsin. There is a condition where there is deficiency of this alpha-1 antitrypsin. Sometimes due to smoking, these free radicals can be produced. These free radicals can inhibit alpha-1 antitrypsin. That is called centriacinar emphysema. So that this elastase will digest the alveolar wall. So there is abnormal and permanent enlargement of the alveolar wall or destruction of the alveolar wall takes place due to smoking. The free radicals inhibiting alpha-1 antitrypsin and the elastase digesting all the alveolar tissue. That is called centriacinar emphysema. Then there is panacinar emphysema. In panacinar emphysema, destruction of alveolar wall occurs due to deficiency of alpha-1 antitrypsin. Sometimes alpha-1 antitrypsin is produced, but it is abnormal. These abnormal proteins are not transported out of the liver. Liver synthesizes this enzyme. So there is panacinar emphysema. Okay. In panacinar emphysema, which is the region predominantly affected? Panacinar emphysema predominantly affects the lower lobe of the lung. Why lower lobe of the lung? We know that the perfusion and ventilation is maximum in the lower lobe. Okay. As we go down from the upper to lower part, both ventilation and perfusion increases. But the perfusion increases more than ventilation. So if you take a ventilation perfusion ratio, it is more at the upper lobe. That is why TB affects the upper lobe. Ventilation perfusion ratio is more there. So that tuberculosis affects the apex of the lung. But both ventilation and perfusion is more at the lower lobe. So because there is more perfusion at the lower lobe, more neutrophil reaches at the lower lobe. So there are these neutrophils producing elastase enzyme will digest the alveolar wall. Why? Because there is no alpha-1 antitrypsin in panacinar emphysema, either because the deficiency of alpha-1 antitrypsin. So panacinar emphysema affect the lower lobe. So these are some of the things we need to know about the neutrophils. Okay.